So thank you very much, Tom, and the Mycological Society to have this great opportunity here to speak. My presentation is quite large. It will be almost 200 slides, but some will be very fast and some I need to skip, or we will see. I hope I speak 1.5 hours, perhaps a bit longer. This presentation was already given in Amsterdam in 2015, but only a very short version because I had only 5 minutes and I needed 10 minutes then. And in December 2023, in last December, we gave the whole presentation here, but in German. The reason was that we got the Antoine de Paris Prize from the Regensburg Botanical Society, because Professor Brzezinski recommended us for this. So we go to the first slide, and you see here a picture of Western Australia. This is one of the collecting sites, one of the many collecting sites. Mediterranean landscape, and this path illustrates the endless way we are going in taxonomy. We never will reach the end. The monograph is about 7 kilo heavy, written in DIN A4, 2 volumes. And you can see here that you can download it as a PDF. And also I intend the presentation to be able to download. So you can later have a look at special things. Some slides are very overloaded with information, and I cannot address all things that I wish to. There is also some German text because of this German presentation. There are over 1,700 pages, over 1,000 plates, 94 tables, and over 70 phylogenetic trees. The 3 authors I was already introduced. The issue with the Sarcus cipher, which Tom addressed, was a case study for the Weitzer taxonomy. But my professor, he wanted that I do a study on another group to prove the importance of this method. And at that time I choose Orbelia because I knew 12 species, and because of the spore body, which is only visible in the living state, it was a good idea to make such a proof, and it should become a small paper. And finally it took decades of years, because the monograph took about 35 years until it finally was published in 2020. Effie, my best friend, she studied in Regensburg the polyploidy of discomycetes, and because she had problems with the identification of her material, we came in contact, and it was the first time that we met. And so Effie soon worked with the Orbelia, because I started the third person, Guy Masson. He soon joined to this this project, and Effie later. Effie mainly worked with the cultures of Orbelia. She tested different animals, because Orbilia is known to catch animals. And Guy was the best collector in this study. Without him, most of the species would have been overlooked. And later he concentrated on the DNA sequencing, also culturing. But his profession was an engine driver and a metal worker. He's very skilled in making tools like hand lenses, electric hand lenses, or my macro objective was built by him. This work would not have been possible with the many people who contributed to our monograph. To do such a monograph needs several things. You must have an idea. This is mainly the vital taxonomy. This offers a huge amount of new information. Another prerequisite is that you must have a lot of time. Usually people have not the time when they are working on the university. They are not very long in the university. And what I needed is to work even at the evening, at the weekend. I'm doing this without I have no employment. It's a private study, and of course this requires a special lifestyle, which includes an old house which we own, no costs for the firewood, to have no car, and so on. I should also say that sequencing in this work was done by Michael Weiss in Tübingen and Gregor Hagedorn in, I don't know. The photographs in this presentation are not given with I must excuse, but you can see them in the monograph. They are mentioned there. Now an overview about the presentation. We start with an introduction. Then the teleomorph and the anamorph is dealt with on the morphology, also the genus and species concepts. Then I will give examples of the 10 accepted genera, examples with problematic interpretation, a chapter about vital taxonomy, a chapter on trapping of nematodes and insects, and finally, I think the nicest or relaxing chapter is the ecology. So let's start. An overview on the taxonomy. The class Orbiliomycetes was described in 2003, so rather lately. Earlier, it was a family of the Helothiales. And I remember my professor Oberwinkler, he was not very excited of the decision to raise a family to a class of Ascomycetes. He could not imagine that this has a good background. But the background was, on the one hand, the DNA, and on the other hand, the vital characters, mainly of the spore body, which is unknown in any other group of Ascomycetes. So we have here 10 genera in one family, and you see the largest. I will use the cursor now. The largest genus is Orbilia in our concept, at least, with over 400 species. Also, a large genus is Hyalobilia. Out of these genera, 6 we have described as new. And you also see that not every genus is known from the teleomorph. I just see a mistake. The Amphosoma is only known from the teleomorph. No, it's, well, no, it's no mistake. When there is a red sign, it's only known from the teleomorph, and when it's a green sign, it's only known from the anamorph. We have here figured a basidium, so for people who have little insight, the ascomycetes, in German Schlauchpilze, and the basidiomycetes, in which the spores are exogen formed. Now the position of the orbiliomycetes, the reason why we have recognized the group in a separate class. You see here below the basidiomycetes, on this spot, the asci came into function, and you see here two low groups which are characterized by the absence of paraphysis. They have asci but no paraphysis. Then we are here in the green bottom, the orbiliomycetes branch off from all the remaining ascomycetes. And we can have here the amyloidity of the ascus. This means that amyloid apical rings occur for the first time, but also amyloid outer layers of the ascus. But I must say that in Neolecta, Taphrino also occur hemiamyloid outer wall layers. But the apical ring is never seen in the orbiliomycetes, and the orbiliomycetes have always inamyloid asci. This means that they probably derive from an ancestor which did not yet develop an apical ring. Here I give the present knowledge. It's a bit, it's not up to date, but rather the blue figures illustrate what was known before the monograph, and the red figures tell us what we have now observed. And these differences, for example, between North America and South America, they are surely not real. It is only that we have had few collections from South America, also from New Zealand. There surely exist much more species, like, for example, in Australia. And the reason for the large number of Australia and North America are the different expeditions which Guy Masson made during this monograph. Also, Chinese workers were very active, and of course, this were in Europe. Many many workers have collected and supplied us their collections. In Africa is also very under-collected. Here you see another illustration in the upper row. You see the abundance of the collections, and in the lower only the spots. And this is also not up to date, because New Zealand is without any spot. This is no more true. Now come a few slides to give you the idea how these fungi look. Orbilia xanthostigma is the most often collected orbilia, but it is not. It is very, very frequent and all over the world. But there are many other species which are similarly frequent. The reason I will tell afterwards, but I give a hint. The word hykrik means that these fungi grow on the ground, on the moist ground with a permanent humidity. And this is also true for the next one. It was earlier also placed in the genus Orbilia, but now it is Hyalorbilia we recognize in a separate genus. And here's another one which grows on the ground. This was separated in a different genus, Hyalinia, and the epithet crystallina. And when we transferred it to Orbilia, we had to change the name because there was already an Orbilia crystallina, which was an operculate fungus. This is characterized by teeth on the margin. Now, this is a species which is inhabits a xeric habitat. It means branches that are still attached to the trees. So these fungi tolerate more or less long periods of drought. And again, a name change. Orbilia coccinella was the old name, and this was in use over a long time. Then it changed to Orbilia alnea, and we changed it to eucalypti because coccinella, we will have it later. It's a totally different species. It was a misidentification. And here is another example for a xeric habitat. Again, a name change, Haprostictis rubra, a separate genus was recognized, but we transferred it to Orbilia and used the older name Carpopoloides. In the meantime, we think that there are two species, two very close species. One is rubra, one is Carpopoloides, and there are also more of this close relationship. Here come two slides which are sorted after the habitat Hygric in this case. So you see the Hygric species are often hyaline or almost so cream colored. Exists a brownish one, a deep red one, or yellow. And sometimes a subiculum is visible, especially when the apothecia get dry and contract. And here we have Orbilias from the xeric habitat. They are often more brightly colored, orange, yellow, rose colored, and even deep purple in this species, or brownish. And we have also some black species. And among the drought-tolerant species, many species are such small, and when the substrate is dry, you almost don't see them. These are all hydrated apothecia, and they are all in the living state. Now, why is this book so heavy? It was the exponential increase of the species which we have detected. And the reason for this increase is I don't know what this disturbing. I'm not sure if it is still working. No.
Try and do the share screen again. For some reason, we lost the presentation.
I get a window above and disturbed. So this upper row shows symbolizes the vital taxonomy, which means we have spore bodies, we have also these bodies.
We're not seeing the presentation anymore. Can you try and press that the green share screen button?
Yes.
The bottom again. There we go.
Mm-hmm.
Great.
Okay. This upper row shows symbolizes the vital taxonomy. We have the living apothecia, spore bodies in the spores, these cytoplasmic bodies, and here we have a herbarium taxonomy. The color of the apothecia disappears with age. This was also reddish when alive. The spore bodies disappear. The paraphyses lose their contents. And the one reason of this increase is that most species are drought tolerant. They are collected on these exposed dead substrates. People often are used to collect on the ground, and they overlooked a lot of species. Now I give some hints on the morphology and the genus and species concepts. The apothecia with the extipulum, I cannot speak too much about this. Interesting are the hairs at the margin which are partly with glassy processes on their tops, characteristic for species which then also have teeth at the margin. Spore bodies have different shapes, very important for classification. An important thing is that the spores are partly inverted within the asci when they are formed. So we see the spore body pointing downwards and pointing upwards. This cytoplasmic body is called SCB, soluble because in KOH it disappears, cytoplasmic body. The exudate has different amounts, and we have different shapes of the asci, thin-walled, thick-walled, or such truncate, depending on the view. And when they open during spore discharge, they get always such a split in the apex because they have no ring. Here I give a short idea how I make my drawings. I make it freehand and I measure the spores. I put such 4 points and then I draw the spores in. So it becomes at 4,000 times I made the spores. I say 3,000 times. And finally I must, the drawing looks like this. And then I make the fine drawing, the finished drawing directly on the paper. Woodier was a famous vital taxonomist. He studied only fresh collections. And here you see the frequent xanthostigma and the coccinella, which is now eucalypti. And you can notice also he studied only the living spores. You cannot see a spore body in his drawings. This is difficult to explain, but mainly you always find dead spores in a preparation and you must disregard them or you must study them separately from the living spores. And Boudier did not notice this, only in one case, but he did not publish that. And here two other plates of his book, which just before we had these pieces already. You don't see the spore body. Also here, no spore body. This one has lanceolate, paraphysis, apices. For this reason, people have separated the genus Haprostictis. And Hyalinia was first defined by uninflated paraphysis, in contrast to the knob-shaped paraphysis of Orbilia. This knob shape was overestimated because it occurs also in Heliochaelis. But it is quite a typical character in Orbilia, but not in many species do not have it. And later, the genus Hyalinia was redefined based on the margin with the teeth, with the glassy processes. And Hypostictis was also defined by such lacineate margin, and it was long placed in the Dermatiaceae. It was not recognized that it is related to Orbilia. The spore body is an old thing. It's not my detection. There were three French mycologists who already saw the spore body. You see, it is over 100 years ago. The microscopes apparently have been good enough to see this structure. It is very conspicuous, but it disappears. You see here in Patouillard, he's had one dead spore, or here also, and these are living spores. The dead spores get such granulated, and Patouillard even saw that the spores are the asci are 16 spored. There are 16 or almost so spore bodies inside, but since he did not see the spore wall in these dead asci, he overlooked, he wrote that this species is finally 8 spored, which is not true. This is a 16 spored species. And here, I think in this species, Roland use some fantasy, because the spores on the lower spores of Nascus, they are almost always inverted. The inversion of the spores is visible in Patouillard's drawing. There are spores which point with their acute end downwards, and these are inverted. And also Chenantais overlooked that spores are partly inverted inside the asci. This is a phylogenetic tree of Orbilia or Orbiliomycetes. The large genus Orbilia, we have not divided it into several genera for some reason, while the other genera here in this tree, not all genera have DNA. They are rather small and are located at the bottom. And within Orbilia, we distinguished different sections, which you can see here, of different size. And you see also, it is a maximum likelihood tree of these DNA regions. You see that the section Orange-Rubre is from the molecular view heterogeneous. It forms three clades, but nevertheless we decided to recognize one section. You never know when you do multi-gene analysis if it changes this arrangement, and we also thought it is better to have a morphological concept in the first place and to use DNA as a confirmation, or sometimes not confirmation. Also in this Hemiorbilia, we have here 2 clades. And now this tree will appear several times on the next slides. One character of Orbilia is the ascus base. It is often such variable, but without acrosia. Simple septate ascus bases are typical of Orbilia. Illustrated here with a minus, and these characters, T is for the T-shaped, L is for the L-shaped ascus base. in the genus Amphosoma, we have both simple septate and croziers, depending on the species. In the Lilapila, we have croziers with a perforation. Also in Hyalobelia, we have croziers in all species. So the ascospace is obviously a good character to for the phylogeny, for the investigating the phylogeny. The spore bodies are of similar importance, but there are some convergences, so that it's not always easy to recognize the group from the spore body. Interesting is that some species have two spore bodies, one at each end or more, so that the spore is homopolar in the content, also here. But others have only one spore body, and this is always at one end, which I defined as the upper spore end. It's without the spore body, you sometimes do not know which is the upper or the lower spore end. But when the spore body is there, when you have it, you know. So we have there elongated spore bodies, round spore bodies, rod-shaped or such lantern-shaped with a fine phylum. Lens-shaped is a very striking feature, which is also in Lilapila, very similar developed. Phylogenetically, these homopolar spore contents are only found in the Hyalobelia and in Amphosoma, which we named after this character. But Hyalobelia has also species with such single spore bodies. Here's another tree which illustrates spore shape. It also includes series. We have here only with this color only the sections, and here we have also written the series and nematophagy, which is known here, and also here, and also here. But no, that was not true. I must go to the next slide. The spore shape was used as a taxonomic character also at higher levels in earlier times. Svirchek tried to make groups according to the spore shape, but this turned out to be polyphyletic. Much more important are the spore contents. For example, in Hyalobelia, we have these homopolar spore contents. A very similar spore is in Arthropotris, in a section of Orbilia, but you see there's only one spore body at the top. Or in section Luteorubelle, we have such arcuate spores, or in section Hemiorbilia, or in Haprostictis. So this is often very closely related species, differ in the shape or in the degree of curvature. Also, these reniform spores occur in different groups. These subglobospores in very unrelated groups, and also spores with a tail. They are not related to each other. The classic processes which were used for the genus Hyalinia occur in various groups. So it has little taxonomic value between species, yes, but not much more. The paraphyses, whether they are inflated or not, or have different shapes at the apex, they occur across the group. Little correlation with the with a relationship. The number of spores. We must say that polyspory in Orbilia was unknown prior to this monograph. We have detected the first species and quite a lot of polysporous species we have. And you see some groups are constantly 8-spored, others are frequently polyspored, but also 8-spored. So this is a character, a polyphyletic character, but of some higher level importance. And all species which are polyspored occur in xeric habitats. No, not a single species which is intolerant against drought. An illustration of polyspored asci, the spore numbers are similar, the spore shape very different, also the spore body is very different. 137 species were polyspored, which we have in the monograph, about 28% of all species. Now the cytoplasmic body in the extipular cells and the paraphysis, a very striking organelle, which we do not know what is the composition, and it occurs across the group of the genus or even the class, but it is absent in some groups. So you here see the diversity of ascospore shape and spore bodies. The lens-shaped spore body can also be get such an elongation, and related to these species are also such in which the attachment to the spore apex is very narrow, becomes narrow, and also these ones, they form a group. Hyalobelia forms a group, and here you see the spore shape diversity in the genus, in the subgenus Hemiorbilia, species with an apical apparatus or apical thickening. Now, there are many species which have spore tails, and the reason why they have such a tail is unknown. One could think that these species, these spores, have an aerodynamic function when they are forcibly ejected, but this can easily be disproved because the spores, also such spores, are partly inversely oriented in the assae. So this makes no sense for aerodynamics. Rather, the spores in every Orbilia, when the ascusor attains full turgescence, they form a packet. They form a single body by gluing together, and then the tail has no function at all. I suppose the function of the tail comes later in the life cycle, but we do not know. Spore bodies can also be viewed in the electron microscopy, but in the herbarium material you have problems to see them. Here are two examples where I saw the spore body in holotypes, in old holotypes, but less than on this drawing. It is very faintly, you see the region, and also the shape changed. You have a empty space here, and you are lucky to see the orientation, because these two species are closely related and easily confused, and the main distinguishing feature is the orientation. The upper spores oriented downwards and the lower upwards, and here the reverse. So I could see this feature in the types, and therefore I could apply the names. These workers here, about who identified Lutherobella, but I think it's Sarraceniana because of the spore orientation. This is a crop of here, of the middle of the ascus. Illustrates these workers, they were unaware of seeing the spore body under the light microscope. They saw it only under a transmission electron microscope because they made their preparations in lethal media. They used Melzer's or cotton blue, lactophenol or something, and in such preparations, the spore body immediately disappears. Here are light microscopy photos of the spore body. The spore body is a vacuolus structure. When you add cressyl blue, aqueous cressyl blue to the preparation, you get a selective accumulation of the dye in the spore body. So this is one proof for their vacuolar nature. The cytoplasm also takes up the dye, but it doesn't accumulate. It is typical for vacuoles that certain vital stains are accumulated inside vacuoles. Here is an electron microscopic photo. It was made by Rosanne Healy and Don Pfister and presented in Amsterdam in a lecture. And this photo is so nice that I decided to show it here, because in this direction we should continue research. The spore body is always attached to the apex of the spore. Here in a very broad manner, sometimes only as a very narrow phylum. And we do not know what is in the spore body. We do not have an idea for what reason Orbilias have this spore body. But we see here that such tracti or canals that go from the cytoplasm into this vacuole, and this way normal vacuoles also work. They have such canals which lead substances into the spore body. I guess this substance in the spore body is formed in the cytoplasm and is transported here into the spore body. When spores have their spore body on the lateral side instead of the apical end, it becomes difficult to speak of an upper and a lower spore end. But it seems that the spore body migrated from the apex to the sides in the evolution. Here in this spore, we can imagine that this was the upper end and this was the lower end. At least we recognize a heteropolarity here, more or less also, but sometimes not. And here it's also becoming difficult to say which is the upper and the lower end. The orientation of the spores in the ascus is irregular. And also in this species, we have an irregular orientation. It's a funny species. We called it Corculispora because of the spore body, like a heart, a small heart. Spore body lateral. You have no chance when the spore is dead to say which is the upper and lower end. And also in the living state, you don't know because the spore body is on the side. But sometimes this species forms such aberrant spores, and some the spore body is more at the apex, or it is not clearly at the sides. So this is some kind of proof or hint how the spore body moved during evolution. I'm not sure now why this slide comes. There should have been a heading about the anamorphs. Now we arrive at the anamorphs. Orbilia has, I think every species has an anamorph. The apothecia are usually collected, but some people, they collect only anamorphs. They are fixed to the anamorphs, and the relation between anamorph and teleomorph was mainly detected by Don Pfister, who made the first proofs for their relationship. So with a good dissecting microscope, you can see the anamorph. It is hyphomycetus, it has conidiophores and apical conidia, which may look like this and rarely occur in such abundance, and sometimes you can find them only by scratching on the substrate. In culture, the orbilias grow more or less slowly. The intolerant species grow faster than the tolerant ones. The tolerant species often have such a reddish color in culture, and some make such repetitive growth. Conidial shape is very diverse. There have been described a lot of genera for these different conidial shapes, and this is not phylogenetically proved. It was an artificial system. There are branched ones and unbranched ones, and usually they are septate, but there exists also a non-septate conidia more rarely. And the terminology about these conidia is very complicated, also how to describe them and for what reason they are adapted. It is a very interesting topic. These Trinacrium conidia may occur in different relationships. You see the sections. They look quite similar, but they in the same group occur also unbranched ones, or even different shapes. So you cannot define a group by the conidial shape in this case. In a smaller group, the section Haprostictis, we have very, very similar conidia, almost identical, but the spore shape and spore bodies are distinctly different. So we can distinguish species by the spores, but not by the conidia. Now we come again to the phylogeny from before and have here the different conidial shapes. These ones with aseptate conidia with protuberances, they are very exceptional. We first did not believe in them. We thought they are contaminants, but they belong in this group where otherwise such helicoid ones and Phragmosporous conidia are typical. Within the nematode trapping fungi occur a variety of conidial shapes, but rarely they are branched. And again here, the name Trinacrium was used, although this is a very exceptional case and all the other Trinacriums are unrelated to this. Again, a picture about the conidial shapes. I think I must go further. The Trinacriums, they occur also in Amphosoma or in Hyalobilia. So Trinacrium is everywhere. Now a slide on the nematode trapping fungi. This is this group whereby the Dactylella is without trapping capabilities. And within these four groups, we have different trapping organs. The phylogeny, the monophily of these groups are defined or correlated by different trapping organs, which are very famous and long known. And it is funny now to connect these anamorphs to teleomorphs, or at least to the relationship. Here is an example of two species which I cannot, or very difficult, cannot distinguish from the teleomorph. They have very similar spores, very similar apothecia, grow in wet habitats, partly submerged. And one makes this helico anamorph, and the other makes this angulospora anamorph. In principle, these are almost identical conidia, but they are the degree of curvature is the distinguishing feature. And when they are such curled, they trap air inside the conidia. The reason is that these conidia can swim on the standing water, while these conidia are adapted to the running water. And in fact, these two species are usually found in different habitats and explain the adaptation with their conidia, or the function of their conidia. They are transported by the water. And I think just in the standing water, there are animals involved, such birds also, which carry the conidia to another place. It was Don Pfister who detected for the first time the connection of helicoon with Orbilia luteobella. And we could confirm this connection by pure culture and also by DNA. But in Pfister's work, there was a mistake that the cultures were contaminated, and the DNA which he found and put in his trees belonged to a hypocorallian species. So for a long time during our monograph, we doubted this relationship until we found it by ourselves and confirmed it. And in the same group occur these strange conidia, which I shown you before. The conidial shape is so similar that the species can hardly be distinguished. With them, better are the spores, which show a bit more characters, but this group is really difficult in species definition, separation. And at first, all these conidia we knew only from China, from Asia, and we thought they do not occur in Europe. But now more and more finds exist which clearly show that these occur also in Europe. We had a recent case where we could prove also by DNA that the same species occurs in China and also in Europe. The genus Pseudotripoconidium was created for these conidia by these two workers, and we put this genus in. We put it down to a series of Orbilia. We had a lot of contact with them, and they sent us apothecia for documentation, for identification. So many of their collections are in the monograph. These different conidia, they are also recognized by people who investigate the trees, living trees, for their conidia funga. When rain is falling on the trees, they collect the water in holes in bark or wherever up in the crown, and they find these different conidial shapes, and many of them they cannot identify. There are groups which are not related to Orbilia, but there are also many which are Orbiliomycetes. Now the different genera which we recognize in Orbilia. I showed you some black ones. These are only on conifers. And the left one is from North America in the boreal region of the Rocky Mountains. It has 16 spores, 16 spored asci, and the spores have such a thin spore body, for which reason I called it Piliosoma. The other one is 8 spored and is European and has a little different spore body and another spore shape. And it's otherwise very similar. Also, they are closely related, forming belonging to the subgenus Lentiformis. Here an Orbilia which is placed in section Aurantia. It was detected by Guy Marceau on standing beech trees on the detaching bark. You see this bark is completely dry when you go out in the field, unless it was raining, and the apothecia occur on the inner face of the bark. Here together with Orbilia aristata. This is often the case that you have different species on the same substrate close to each other. Another interesting detection was a species apparently confined to Fraxinus, or at least a few species, a few host species, host genera, was detected in Poland by Piotr Perz. This one is from Australia, this multisport asci, spore body attached subapical, strange apothecial morphology, but not consistent, and very, very drought tolerant. You can study such fungi even 4 years after you have collected them, and you can still make cultures from the living asci. Spores are still alive, and a very thick apical thickening. Such curious spore shapes occur in Orbilia here, also from Australia, with a tail which is on the laterally curved, and because of the shape, I called it Myrio muscula. Myrio, this suffix we use for 64 spored asci, exclusively for this spore number. The genus Lilapila was a surprise. At the first time when Guy Marceau detected it, we thought it is a Helothialis because of these hairs, but the spores have a spore body, like we knew also or later from Orbilia. And the asci have arise from Croziers, are thin-walled and hemispherical at the apex, but the strange thing is this dark purplish color. When you try to collect them, they are rather inconspicuous because they have about the color of the substrate. They grow on resin, on the living trees in wounds on black resin, and so you need a sprayer in order to see them in the field, or you go out when it was raining, then it's easier, but you need a hand lens, otherwise you have no chance. And there are different species which are almost identical in the microscopy, and very, very difficult to distinguish. The DNA made the proof that there exist different species. This species is also such purplish, dark purplish, blackish, and it was collected by Fuckel, and it's the only collection which is known, growing on pinus needles. And so my analysis was from the dead material, the spore body I could see in some spores, also the spore orientation I could detect. Capitate paraphysis, but croziers at the base. I described therefore a new genus, but we have no DNA. Hyalobilia, characterized by these mostly homopolar spore contents, have often such colors, whitish, creamish, yellowish. This exudate, this fine granular exudate, is typical on the surface of the hymenium, also on the outside. The extibulum is of a prismatica, parallel to the outside, unlike Orbilia. So the genus Hyalobilia was created because it also formed a very separate clade in phylogeny studies. The subiculum hyphae are often rather broad. This is the most interesting genus, Lecophagus. First, I wanted to describe a new genus, Rotiferodiscus, because the Apothecia, the very minute Apothecia, have connection to rotifers. This is a dry rotifer, which usually revives after rehydration, but this one is dead because it is parasitized by this Lecophagus genus. The rotifers, I hope you know them. They have this organ. When they eat, the material comes in this organ where it is chewed and made smaller to go to the stomach. And this is often, or is sometimes, the only remains of the rotifer where you can recognize it. And here you have it also, but you see the entire animal and the hyphae of the Lecophagus inside. The Lecophagus has very broad subicular hyphae, and these hyphae contain crystals which are also unique in the orbiliomycetes. And with these crystals and the width of the hyphae, I got convinced that the genus Lecophagus, which was only known from the anamorph, is the same species or the same genus and the same group as my Apothecia, which I detected. Here are photos of such Lecophagus species. Here the broad hyphae, Apothecia rather small in comparison to the hyphae, and spore bodies like we know them from Orbilia. This is the trapping organ, is a adhesive peg on which the rotifers they are attracted and think that this is something to eat. And when they are glued to this peg, to adhesive peg, they cannot move away anymore, and the fungus starts growing inside the animal and consumes it. Here a photo of such a species, this musticula. We do not know the teleomorph. We know only the anamorph. And it was sent to me by a German amateur who found it in a pond in his garden on water plants, and made these photographs. Now the genus Amphosoma, which is mainly on conifers, and it has often on wounds, and it has homopolar spores. Characteristic is the vacuolar drops in the paraphysis, which are very conspicuous, refractive like the spore bodies. And inverse spore orientation may also occur here. Also the spore bodies are in both ends. And this one is a monotypic genus rather lately discovered by Enrique Rubio in Asturias. It is growing on soil, a very exceptional situation, and the spores are very difficult to understand. There are spore bodies, but there are also lipid bodies. But we succeeded with a sequence, and only then I got convinced that it is an orbiliomycete, but rather distant to the other genera. Here is the genus Retiarius. It is known to catch pollen grains, and there was no teleomorph known, but in the meantime there exist sequences, so we know the relationship. It is Donat Magyar in Hungary. He made a study on these. The genus Retiarius was described by a South African woman who studied specifically these pollen inhabiting fungi. And Donat Magyar, he described the new genus Mycoceros for another such fungus which catches pollen growing on the living trees on the bark of the trunk. And we also observed sometimes pollen grains in vicinity of the orbilias, so that we assume they are using the pollen for additional nutrients. But in this case, it could also be an egg of an animal, we are not sure. And we made tests with our cultures, and all species were able to invade pollen. So this seems to be a unique feature. Even unrelated fungi, such like Rhizodicina, it invaded pollen in our cultures. Species concepts are often problematic in orbiliomycetes. We have almost 500 species in the monograph, but this is a tentative number. We have a lot of species which we or finds which we did not describe as new because we were not sure, but we separated them and treated them as undetermined. So the real number could be 1,000 or more. Who knows? And it depends also on the species concept. For example, it was a big surprise with the Orbilia xanthostigma. The yellow ones we had in the beginning, but there exist also white ones, and they even grow mixed together. So I was sure this is only an albino. The spores are the same here from the white ones, here from the yellow ones. All microscopy was the same, only the yellow pigment was lacking in these white ones. Now it took some time until we had the opportunity to get sequences from them. The white ones are not so frequent. It's not so easy to find them. So, but there was finally a log which had on the upper side yellow ones and on the lower side white ones, and the yellow ones also on the sides, but they need a light. They do not grow hidden on the lower side of a log, while the white ones grow everywhere, but mainly on the lower side. So from one collection, we took from the yellow ones and from the white ones a DNA, and the result was very, very striking. The distance between the white and the yellow ones was 16%, 16.5% in the ITS, which is usually seen between different genera, or at least between different subgroups of a species, of a genus. So this was very difficult to understand, but finally we acknowledged it, and we did not separate the two species in the monograph despite this high distance, because this was rather lately in the process of the monograph, and it would have caused a huge amount of work. So we left it as one species. Here you can see these two species are the first species which were described by Fries, and he described them in Petitza. And later, many years later, he described the genus Orbilia and combined them in the genus. Here the situation outside where Orbilia xanthostigma is collected. Here again, a mixture of white and yellow ones. I'm sure these are different species, not the same. And when you look at the same area in the herbarium, the color, it is rehydrated. In both cases here, the yellow color is still there, but it is less conspicuous. And sometimes it is even difficult to be sure where they're yellow or where they're white. So with the old material, you have not a good chance to determine the leucostigma or xanthostigma unless you succeed with DNA. Now, the situation of this xanthostigma-leucostigma complex is much more complicated when looking all over the world. We were in Europe with the detection of that they are two species. Fries also collected in Europe, and this clade above is xanthostigma. This clade here is leucostigma with much less sequences and collections. In America, we have also white ones, but they are different genetically, although the microscopy, they are the same. And we have in North America also here the yellow ones, but they are different from those from Europe. We have in Europe one collection which deviates. It is more tolerant to drought, microscopically the same. We have several clades in New Zealand. The microscopy is the same. You cannot identify them with a microscope. But there are also this Xanthoplexa. It has smooth ascospores. These all had warted ascospores, but this Xanthoplexa has smooth spores. The Aureogranulata has also warted spores. It is more or less recognizable, and it is tropical, but heterogeneous also. So in this case, we do not know what to do. We did not describe any of these. We did not describe new species. Also, these clades are very well defined. Some people doubt if ITS is a valuable gene region, but our results confirm this, because we have never seen something else. We have a very consistent ITS within Europe, and also usually within in the other continents. This is all xanthostigma, or perhaps leucostigma. Only this number 5 is in the living state. The others are in the dead state. Published. No, it is a re-examination by me from type material. Person had already a name for them. Aurea he used. Personi is a synonym of xanthostigma, but it is from Chile. So from South America, I guess it is a different genotype, but we do not know. It's a very old collection by Montagne. Here is the holotype of Rufula from Pennsylvania. I think it's from Schweinitz. Also, I identified as Xanthostigma. And here is the holotype of Delicatula by Karsten. So this is a European collection, and it is very surely Xanthostigma in our sense. Belenovsky, he had the name Paradoxa. It's a new species, and he overlooked or he saw the spore bodies, but he saw two spore bodies in each spore, and I'm sure this was is an error. It was a bit of fantasy. This Xanthostigma has such an anamorph with this type of conidia, and a very similar anamorph is known in this case, Petilospora dactylopaga. I have the idea that this species, which traps amoebae, described by Drexler, is at least that it belongs in the group of the Xanthostigma leucostigma complex. But evidence is lacking because I'm not unaware of a culture of this species existing in culture collections. Eucalypti is now I'm a bit disturbed. Yes, Eucalypti is a species which is very frequent and variable. It exists in a small sport variant, in a large sport variant, and all intermediate cases. And it grows on the ground and on in the xeric habitats usually. But for a long time, we had no DNA at all. And later, more and more DNA came through the activities of Guy Marceau. He sequenced a lot. And we do not have morphology regrettably for all these clades, but whenever we find no clear correlation so that we can say this one is defined by some kind of morphology. Here are conidia a little different than those, but in the end, we have a lot of clades and only one species. In Orbiliomycetes, the ITS is very diverse, meaning high distances between the species, even more than we know from the Helothiales. So with ITS, you can easily make taxonomy, but in some cases, this is a most extreme case. You have no idea what this means. These species below, they are also very similar, like this Eucalypti, but we finally could separate them with morphology, more or less. And this is the type of Orbilia coccinella. It is a species with reniform spores. It has 16 spored asci. It is a drought-tolerant species, and it is only known from this single collection, very old collection by Sommerfeld in the very north of Norway. People tried to find it again, but so far failed, and we have no DNA, but we know it is not the same as Eucalypti. Eucalypti is the type of the type comes from North America. Orbilia euonymi is a 32 spored species, but it was described by Belenovsky with 8 spores. The type I studied and found 32 spores. So Belenovsky was unaware of the multispory of this species. The distribution, the species is very frequent. These dots, they represent the collecting activities. The species occurs everywhere in the temperate or Mediterranean region. Finally, we found that different spore numbers exist, and from 16 to 128. And at first I thought, this is one species, these are not different species, because we found this 128 also in Europe like this one, and I could not believe. But when we had DNA, we saw they are different. The 32 spore species is the most frequent, and so we have most sequences are from it. And the 64 is only known from Australia and China. But the 16 spore is also in Europe, and now and then we found it. This is another the only other case of a species described in the literature with more than 8 spores. But again, Feldgen saw only 8 spores in the ascus. Incredible, but there is no doubt that this is the same species as what I studied from the type. The type is rather small with a few apothecia, and the spore shape is the same, spore size. So he simply overlooked the polyspory, the 32 spores. In the dead state, I saw also the lens-shaped spore body, but it looks different like in the living state. The xeric habitat means that the apothecia are shrunken and often almost impossible to see. You must spray them with water to find them at all. This is in Tübingen, a species which is quite rare, and I detected it at the university, at the old university, on an ulmus tree. Almost the same species was described as Abutylonis by Cash from Hawaii. First, I thought it is the same species, but finally I found, because of this conidium, this single conidium which I saw, it's different from the conidia which we have here. So we decided better to describe the former as a new species, and this one is only known from this dead material. And the third one, which has again very similar spores, had again different conidia, or at least similar like Abutylonis. It is from Mediterranean region and has a different color of the apothecia. So we decided to separate it as a different species, and DNA proved this. These two species are in different genera, but have been confused in the past. Actually, they have been confused. This name Inflatula, no, this name Auriculo was used for apothecia that belong to Inflatula. Such things happen when studies are done on herbarium material. In the living state, you will not confuse them. And if you take care of the crosiers, you will also have the stipitate, asci, and orbilia. You cannot confuse them. But from the macroscopy, they can really look very, very similar. I must skip because I'm a bit in hurry. 2 examples, one example, 2 species which can easily be distinguished from the spore body, but in the dead state, you will have problems. A little bit with the spore size, but you overlook the main distinguishing feature. In the dead state, you lose all these characters. Even this accumulation of the spores in one packet was completely overlooked, unknown by workers on the herbarium material. The nuclei, the fusion nuclei, you can see in water, simply in water, but the division of the nuclei here, this is my fantasy. Again, in the spores, you can see it. Here, of course, you don't see anything of the nuclei. And it is difficult with a vital taxonomy to interpret what you see. The dead ascus, the living ascus, but the dead ascus contains living spores. Here, the dead ascus contains dead spores. All these cases you must cautiously distinguish. The spores, when they accumulate to one body, they are ejected at one entity, and you see them on the agar as one ball. And sometimes you better count the spores on from such a ball, because here spore counting is a difficult thing. 8 spore species, also the spore body at the apex, not the spore body, the spore package. A preparation, a section of a living apothecium, but you have dead asci as well. There is a dead ascus because of the razor blade you kill asci. And shrinkage of the asci here in a Heliocealian is enormous in some cases. So measuring asci makes only sense when you say if you have a living ascus measured or a dead ascus. Here another example, the changes from living and dead. Spore discharge here in a lichen, false theories from herbarium workers. This is the real situation. The tractus is easily overlooked in herbarium. Living spores of Hyalobilia with the spore packet like in two over each other, 4 plus 4. And when you look the same species in the herbarium, you see almost nothing, or you can just recognize a little the spores. So collections have often been declared as immature because people could not find any spores, but the collection was mature, in fact. Crocodispora again with the cressyl blue stained. The asci are all dead on this slide, but on this upper picture, also the spores are dead because application of ammonia killed the spores and the spore body disappeared. Even living and dead spores may occur inside one ascus. Here, 3 dead spores and 5 living spores. The SCBs in the living cells we have seen already, and when you add KOH, for example, they disappear, the cells shrink, and also when you heat with a flame the slides, they disappear in the herbarium. You will not see them anymore. My my diploma was on Sarcosypha, and it was because of the lipid bodies in the spores that I distinguished these 2 species, which Boudier distinguished at the variety level. And this Urana is only on Tilia, this Coccinia is always on other trees, on different trees. And there was a 3rd species, Austriaca. And here, the spores of Urana and Austriaca, large lipid bodies, many small lipid bodies. And in the herbarium, you have difficulties because the many small fuse to large aggregates, and you finally cannot distinguish the 2 species. And Legal made her study on herbarium material and concluded finally that Boudier was wrong in distinguishing this Urana. Not only fungi are important for vital taxonomy. Look here is the Spirogyra, which loses its genus character when the cells die, the spiral chloroplasts. This is all in dead state, literature of Orbilia without spore bodies. Example of 3 species which have similar spore shape or size. And when you have the spore bodies, you have very different spore body shapes. And you can even put these three species in different relationships of Orbilia. The nematode trapping group with a different, it was Scholler and Hagedorn who rearranged these old genera according to the trapping organs before the conitial shape was the crucial character, or the conidiophore shape. And this was shown by DNA that this is the correct circumscription, and needed a lot of transfers in these genera, but we recognize them only as series of section or arthropods within Orbilia. And these capabilities of trapping nematodes is interesting in regard to plant parasitic species as a pest control. And the teleomorphs in this group are rather similar, while the anamorphs are very diverse. So we have adhesive pegs, adhesive nets or loops, and constricting rings as the most elaborate type of trap. Here the haustorium is formed when the nematode gets stick to the adhesive knob, and the fungus grows inside the nematode and consumes it. A few species have such a thick gel on the trapping organs, and here these traps are for insects, for larger animals. This collection from Spain, from Asturias, was sent to me, and this photo I also received from the collector. And I noticed these flies here, these dead flies close to the Apothecia. When I put this Orbilia in a moist box over a week or so, I got this strange conidia here. I thought it's the anamorph of the species, but in fact, it is. This is the structure. It is a trapping organ with a big jail around, not like in the usual adhesive knobs, which have no visible jail, no, very thin jail. This has a thick jail, but in the dry state, it is such compressed here. And when you spray with water, it swells to such a globose structure. These are the unswollen. So I first I didn't know what this is, but I got the idea it could be a trapping organ, and so I made animal tests. I put colimbol or a mite or a spider to my Orbilia, and they were immediately trapped when they got in contact with these trapping organs. Only a spider could escape because it is strong enough. Here you see the mite got stick to the trap, and also here this is the trapping organ. The animal got stick with the antenna. And the flies, I also added flies, and finally the mycelium overtops the entire animal, like in the first photo, or the colimbol finally disappears in the mycelium. Trapping of nematodes by arthropods, adhesive nets in which the nematode gets tangled, and the same with the haustoria, which finally the hyphae inside the nematode. Orbilia auriculor with its anamorph, and with the adhesive nets which we obtained in poor culture. The constricting rings are very elaborate, interesting trapping organ which constrict within a second or so when the nematode by accident comes into this loop, this ring. And what was quite unknown is the triangular thickening at the septa. And these are a solid structure, which is also present in the dead state when you look in the KOH. And the Chinese worker managed to have fluorescence with these wall thickenings. The scanning electron microphoto. This is such a species which has constricting rings. This is a Mollissia, and this is the Orbilia, the Polybrocha. And in poor culture, we got these conidia and also trapping organs. Within the section of Arthropotres, the spores look quite similar, as I already told you, but this group is without trapping organs at all. Series Neodactylella, we called it. Dactylellina has these adhesive knobs, and Arthropotres has these adhesive nets. Conical shape is not of very much use, but the trapping organs. We had these spermoides in the dry and the fresh state, or here nanosperma dry and rehydrated. Cryptogena was named after the hidden growth inside the foot surface, and when you spray, you see them suddenly, or here dry and wet, and in the dead state you have difficulties they can look quite similar, but you do not recognize if they are alive or dead when they are not too old, the material. Collecting on attached branches, few people know that. Mixed growth on substrate, you have a stick and you have orbilias on them, and you think this is one species, and then finally you have 5 different orbilia species with very different spores. You have also Halotiales on the same stick. Orbilias may often grow in such beetle galleries, but we do not know why. Here in this case, 3 different species of Orbilia grew in one spot, and I had to microscope almost area of apothecium in order to make this survey on the 3 species, this distribution, this spatial distribution. On this wound, this blackened wound, not only Lilapila and Amphosoma atroolivatium occur, but also an Amphosoma which has whitish apothecia. Here is a Spatium, and this is Cercis. As an example, how to collect these drought-tolerant species here in the Mediterranean region. Bin Liu from China was with us for some months, no, for 5 weeks, for learning how to collect these Orbilias. In the semi-deserts, one can collect Orbilia on these branches. High plains with numerous Artemisia, sagebrush, and you do not know where to start collecting in such an area. You can try to see the apothecia in the field with a lens, but usually you find the apothecia at home. Even in the Death Valley, the Orbilias occur, or here with the Ocotillo and Yucca. Organ pipe cactus. In this case, the substrate may lie on the ground because there it's also dry. Orbilias, of course, occur also in the boreal region of the Rocky Mountains, but not so much species as in the semi-deserts. And in the tropics, the diversity appears to be even lower. It's an interesting experience. Jean-Paul Priou and his wife, they were on holidays in Peru. And when I saw this right photo for the first time, I thought it is fake. Such plant does not exist, but it really exists. Puya raimondi, and on the dead old leaves, an orbilia occurred together with a cistella, which looks very similar. And this orbilia we named Puya, but we do not know if it is restricted to this plant. It's a single collection. This species was from here. Here is Fimbriata from Australia. We first thought it is a Heliotialis, but turned out to be an orbilia under the microscope. Or here, a Mediterranean climate in the southwest of Australia. On Banksia, we have many collections. And here is Jacksonia. Jacksonia was incredible with, I think, 12 or 13 orbilias from one spot on this plant. Macrothamia, a few collections. These two species here. This very nice plant from Australia, Anigoxanthus, an orchidaceae, or I don't know exactly, but a monocot growing with this orbilia, which belongs in the Gamsilella group of Arthropods. It was very interesting, but we do not know the trapping organ. Surely it forms traps, but from the DNA we know where it belongs. Mixed growth on a branch. I often did such a service on what finally we detected on these branches. Or here, Acacia from Western Australia. Again, different species on one host. Or the Adansonia with two species which are already distinguished by the color. This one we had before. It was completely dead regarding the spores and asci, but the paraphysis and the exfibulum was alive. So at least we have partly the living features, but regrettably the spore body is only indistinctly known. Here, this is our house at home. We had a rope for protecting this part of the garden from our dog. This did not work much well. After a winter and snow on it, the rope, finally, I thought there were algae on it, and I thought this would be interesting to look if there could be a fungus on it. And actually, there were different orbilia species, quite a few, and also other discomycetes from other groups. Incredible what on such a rope occurs. Mainly Aristata, but also 5 other species of orbilia. This was the winter situation. The rope carried snow, and
my lecture is still going on.
During
and Carter left. Carter left.
I will soon be at the end. On clothes are orbilias, but this is not specific. On dung, on bones, they may occur, but on dung and bones there is no specific species so far. On polypores is a little tendency. A species which parasitic on an orbilia, a Hyal orbilia parasitizing orbilia, very, very strange. And this is the last slide. Thank you very much for attention. I hope you are still there.
Thank you.
Incredible. Thank you so much.
I'm sorry to have disturbed you for so long time.
Just absolutely fascinating. Thank you so much. We have a number of questions.
I cannot just cannot just move to this
Oh,
presentation.
if you'd like, you could stop your share, and then we'll just have the gallery.
I don't find it. No. It's difficult. Well, I think you hear me, but I don't.
Yeah, you can go up to that upper right corner of your screen where there's the button, and you should be able to do view gallery.
But the screen, I see only Zoom and the gray background. Chat. No. Now I see myself.
Oh, there we go. So now you see the gallery. You see all of us. So we have a number of questions. If you would like to take a few questions, the first one, Danny Newman. You had a couple of questions. Do you want to unmute yourself, Danny? And
yes, okay.
Ask your questions.
Sure. Can you hear me?
Sure can. Nice to see you.
Yeah, likewise. I don't know if I should ask all of them, at least not all at once. I want to give other people a chance.
Yeah, you could ask a couple of them, and then we can come back to you because you did have several.
Yeah. One question I had was how much whole genome sequencing has been done in the Orbiliomycetes?
Oh yes, it's a good question. At the moment, I'm over asked a few, I think, but at the moment I cannot say. I tried with Heliotiales. I tried to extract the rDNA, but with the Orbilias, I think it's only a few. Not ours. We did not do such studies.
I'll ask one other one, then come back if there's time. I think so. Earlier in the presentation, you showed either a section or a series that was phylogenetically distinct, called helicoon, and I was wondering if that implies that group has helicoid conidia, and if therefore helicoid conidia are diagnostic for that group, that section.
But the name helicon was created for this reason, but in our circumscription it includes also the elongated conidia. We took the name because it exists and put it down to a section and series, and it's not clear which helicon species is orbiliaceous at all, because Heligodendron is very, very similar, and Heligodendron does not belong in this group. It is in the, I don't know, it's very different class.
So if you find a helicoid anamorph that you know belongs to an Orbiliomycete, is it?
Also Heligodendron is recognizable from a different sense of rotation. You must try to view the sense of rotation of this conidium. And Heligodendron is, I don't know, right, and others left-handed. So with this, you can, by morphology, you can distinguish. This is a convergence to the same habitat.
Okay. I'll wait to ask the rest of my questions, but thank you. It was an excellent presentation.
We can come back to you, Danny. But Ethan, you had a couple of questions. Why don't you go ahead and ask both of your questions?
Okay, thank you, and thank you, Otto. Excellent talk. I think we can just exclude my question about the nematophagous fungi, because there was after I asked it in the chat, there was a very detailed answer that I think answered every question that I had. But I was looking early in your talk, there was a map of the world distribution of the Orbiliomycetes, and I think I saw that there were a few species in Antarctica, and I was just curious about whether those are introduced in wood from structures made for expeditions, or whether what's going on with that? Is that?
Yes, it's both. There was introduced. There was extracts from wood for houses for their and isolations of, I don't know, cultures or DNA, and others. One is one was a Lycophagus. It was described as Lycophagus antarcticus, but it was made by a zoologist, was the description, and she did not well know about the rules of the botanical code, and so her species was invalid. But Lycophagus antarcticus, it grows there in the water at this on the island at the seashore.
Interesting. So it grows in the water. So the anamorph is what was discovered, or is there a teleomorph for it as well?
In this Lycophagus, it was the teleomorph. No, sorry, it was the trapping organ, and the conidia. Yes, the anamorph. Yes.
Excellent. Thank you very much.
Thank you, Ethan. Roz Lowen, would you like to unmute yourself and ask your question, Roz?
Yeah, I attended a workshop where we extracted spores from streams from the foam in the streams, and many of the conidia had arms extending, just like the pictures of the
Yes.
Anamorphs of some of the species of Orbelia. And I was wondering if those could be anamorphs of Orbelia that are commonly floating in the water.
Yes. Hello, Rose. It's a problem. We have, for example, the genus Descalcia. Maybe you know, it is with like a cross, the shape of a cross with long arms, and it looks very, very much like an anamorph which we have in Orbelia aristata, or this group, and we thought it could be the same, but there is no living culture to make DNA from the type. So such cases exist where we cannot say. We can only judge from the morphology if it is the same. These species are often described long ago, 100 years ago or longer, and so the identification, the convergence is so strong that it is actually in the floating water occur Orbelia species as well as on the rain flow on the trees.
Wow. Well, it was speculated that the conidia could survive because of the increased surface area by having all those weird shapes. So it's just pretty interesting, and of course we did not identify them.
I think the extremities of these conidia are for a bit of floating in the water, but yeah, surely they are drought tolerant, these conidia. So when they are taken by an animal, by a bird, for example, and carried away, the conidia do not get not dead, but they survive.
Anyway, it's an incredible work. Thank you.
Thanks, Roz. Claudette, you had a couple of questions. Do you want to unmute yourself and ask?
Okay, sure. We had two questions because Jonathan is here too, so we each have a question. His question was, the spore numbers are all the power of 2, and would you speculate on the cause for this pattern? 18, 16, 32, 64, 128. That's the
I did not understand well, but did you ask how we estimate or how we counted the spores?
No. It was that when on all the displays you said how many spores were attached to each.
It's my question.
Go ahead. Go ahead.
So the numbers that you counted, they are all powers of 2. So 8 is
right.
16 fourth.
Yes, I did not.
And then you stop.
And my question is, can you speculate why the spore counts are all powers of 2?
Yes, I forgot to say that polyspory can be achieved by 2 different ways. One is true polyspory, the nuclei divide, and it is simply nuclear division from 1 to 2, from 2 to 4, from 4 to 8. Why there are no 4-spored orbilias, I cannot explain. And the other type of polyspory is in that usually 8-spored species germinate in the ascus by forming conidia, and then the ascus is finally filled with conidia, and this happens inside the living ascus in many species of Helocialis or Facetialis or whatever, and then the ascus can easily be mistaken for a polysporous ascus. It is a false polyspory. There is an article about this distinction long ago. So in orbilia, we have always the true polyspory, and therefore we have fixed numbers. I guess I'm not sure in the for example in Dipodascus in this what is it, Saccharomycetaceae, there might be other numbers than 2, 4, 8. I do not know.
Oh, thank you. Thank you. And the second question was, you were describing how they eat phagus or copepods, that some of the fungi consume these water creatures. So I was wondering if when they go if they go completely dormant, when they dry out and there isn't any water, and these are waterborne creatures, do they go dormant the way that a clam or a mussel would at low tide? They're waiting for the tide to rise, for something to swim by for them to catch.
I do not know now what you mean, but my picture showed a rotifer, this orange rotifer. Rotifers occur normally on attached twigs here in the temperate woodlands. They survive drying. They survive in this contracted state, and when it's raining, the rotifer expands. It takes up water and it goes over the bark. The other rotifers which I have shown, they really live in the water.
Okay.
And they yes, I made an interesting observation. Just this Lecophagus, which was sent to me, it survived. The mycelium survived the thaw. When my slide on the microscope, when it's dried down, the animals or whatever there was in the water, they were dead, but the fungus survived the complete dryness. So this is a hint for this Lecophagus that it is occurring as well in a pond, for example, as on the dry branches in the neighboring woodlands.
Okay, thank you very much.
Thank you for those questions, Claudette. Danny, would you like to ask your other question or 2, is it?
Sure, yeah, I think I had 2 other ones. One is related, I think, to one of the questions that was just asked. Are there other prefixes other than Mirio which correspond to different numbers of spores in asci? I think you had said that you reserved the use of Mirio to specifically mean 64 spores.
Yes, Mirio, yes. But this is a specialty of the monograph. It does not apply to other groups. We had the problem to name 500 or to name 330 new species, and you finally have no idea how to name them. So in order to have a good memory to memorize these names, I got the idea to give constant names for the different spore numbers. So we have a lot of Murio species, a lot of poly species, a lot of Pleo species. So Pleo was 16, Poly was 32, and Pleisto is 128.
that these are conventions that you and your co-authors created for the monograph. To your knowledge, it's not a system used outside of that.
Yes, it's exactly. It's only applicable for, or should not be copied. It helps. I think it helps. For us, it was very helpful.
And then my last question was, what species of orbiliomycetes, if any, can be confidently identified without the use of the microscope solely on the basis of macromorphology or ecology or both?
I did not identify by the microscope.
Without the microscope, are there any orbiliomycetes?
Without the microscope, yes. There exist, of course, but it depends on your experience, and you can recognize them. I was often decepted by Hyanorbilia and Orbilia auriculor, or other Orbilias. They may look so similar, and when I make the preparation, then I recognize the genus, not to speak of the species. But surely there exist species, but you must not always think you can recognize them by microscopy. It's more or less a guess.
Okay, thank you.
Thanks, Danny. Anyone else have a question? If you do, please feel free to unmute yourself and ask your question. Anyone? Anyone?
Hi, I have a question. Can you hear me?
Yes, is that Jason?
Yes.
Hello.
Hi, Jason.
Hi.
Please
Hi.
ask away.
Thank you, Otto. I had a question. You said you did your drawings freehand, and I always assumed that you used a drawing tube because they were so incredibly precise and detailed. So do you use a drawing tube, or are they indeed all freehand?
I'm not acquainted with drawing tubes. No, I don't have one, and I tried a little when I was in Guy Marceau's home. He had one. He used it, and I know how it works, and yes, but was not of use much for me. It needs some skills, of course, this freehand drawing, but the exactness is not. Yeah, it's difficult to explain. You must be more or less the proportions must fit you, but it's really you can also combine a little from different asci, the spores inside the ascus. You combine it a little bit, but you must show the relevant features, and this is with a drawing tube. And the spores are floating, for example. You cannot fix them. When you make vital taxonomy, you can not use a liquid which keeps them from moving. So we must live with the floating spores.
Thank you.
Yeah,
thanks, Jason. Yes, Roz, please go ahead.
I have another question. How often do you find the anamorph when you're collecting, or do you have to bring them into a moist chamber to grow the anamorph?
Guy Marceau, he developed a method with the water drop, with a glass rod and the water drop. I'm sorry, I did not show. It's so much things about this glass that you cannot cope with 2 hours. The glass rod, you put a water drop on the bark close to the apothecia, and then you take back the water on a slide, on a dry slide, and this you will repeat to collect free conidia on the bark. And this way you can find anamorphs, but of course not the conidiophores, only the conidia. With this method we have a lot of observations, but you are not sure if the conidia belong to the species, because they grow intermingled. But the anamorph with conidiophores we saw rather rarely. It's really an exception, and not in the tolerant species almost never.
Well, so then do you try to do DNA on the anamorph?
DNA from the anamorph, only from culture. I'm a little bit over asked. Usually we took apothecia and took the DNA from that. And in quite a few cases, we have the cultures and took the DNA from there. Or this was mainly made by others, not by us.
Interesting. Thank you.
Anyone else?
I'm going to be greedy and ask one more question.
Please, Danny, go right ahead.
I was surprised to see that there was anything other than tap water that you could use and still see living tissue or specifically spore bodies. So Crescel blue being able to be used as a stain and have that accumulate in the spore bodies. I was mistakenly under the impression that if you introduce basically anything but tap water, that you'll kill or risk killing the material. Is there anything other than Crescel blue that can be used in vital taxonomy that will keep the tissue living?
First, the Crescel blue must be aqueous. It may not contain any ingredients other than Crescel blue. It exists also in such a variant. You can use every vital stain, such nitrile red or toluidine blue is just the same as Crescel blue in its behavior. And you can also use Lugol for vital staining, but it doesn't stain much. It enhances a bit the contrast. You can stain glycogen bodies in the spores, which get red. But it is toxic, and after some minutes, you must be aware when the cells die. In Crescel blue also, it is also toxic. It takes for a while, but for a longer time, it will not survive.
Okay, thank you.
Thanks, Danny. Now, Sigrid had posted links in the chat to the 2-volume monograph on Orbiliomycetes, and also the link for this presentation. So if you're interested there, you can look for them in the chat. I also just put in the chat the link to Zotto's homepage, which yes, if you have not visited before, you really should. And also, I would think that most of you are familiar with ASCO France. If you're not, Zotto is very active on ASCO France, and it's a great resource. So if you aren't familiar with it, I encourage you to visit it. And then finally, there is the website ascomycete.org. And if you're not familiar with this, it's well worth a look. And yes, Jason, Sigrid did post the link to the download to the 2-volume monograph. So that's in the chat. Zotto, thank you so much. This has been amazing. It's we're really ending this year's series of talks on a high note. I can't tell you how much I have appreciated this talk. Thank you so much.
I thank also very much for this opportunity and that so many attended this presentation. Thanks a lot.
Yeah, and thank you all for coming. I look forward to seeing you all again next year. Thank you so much, Zotto. Thank you so much. An amazing talk. Thank you.
Thank you.
Great to see everybody. Thank you. Hope to see you all again soon.
Yeah.
Bye-bye.